Yes. So all of us know desmids are protists. So they are unicellular and eukaryotic. We have certain examples under desmids and certain unique characters about them that we need to learn gypsum crystals in Clostridium. As you can see, we can see Clostridium in the second example here, and this is not anything which is an algae or something. This is a desmid, a band like structure, and it has gypsum crystals. What is gypsum? Gypsum is calcium sulfate, and at the tip of semicells. This point has to be elaborated little. So if you see, the Clostridium is a chrysophyte because it is a desmid. For me it looks like a thin cucumber, and what I drew is cell wall, siliceous cell wall, and inside the cell wall I have the plasma membrane, and here the cytoplasm divides into two compartments. Two compartments, 1 and 2, which is joined by a bridge-like structure called isthmus. The word isthmus means bridge. You have an isthmus in thyroid gland, we have an isthmus in the liver, we have an isthmus in the Clostridium. So this is a communication between these two compartments. I want you people to note down, it is cytoplasm which is divided into two compartments, not the cell. One cell. But the cytoplasm is divided into two compartments. Each compartment is called as semicell. Semi means half, no? Half of the cell. Semicell. And at the tip of the semicell, if you see, this is the semicell. I am not drawing the cell wall here. I drew only one semicell. At the tip of the semicell, we have vacuoles. These vacuoles are called as terminal vacuoles because they are present at the tip. And inside the terminal vacuoles, if I enlarge this one, inside the terminal vacuoles, I see calcium sulfate crystals. The gypsum crystals showing Brownian movement. I think all of you know what is a Brownian movement. The random movement of a molecule. Brownian movement. So we wonder, why should we have calcium sulfate crystals in the terminal vacuole of a semicell in Clostridium? This gypsum crystals acts as statolith. Statoliths means crystals which will sense gravity. You know this. Gravity. Okay, because these are denser than the surrounding fluid in the vacuole, they tend to sink to the bottom of the vacuole, sensing or suggesting gravity is operating there. So this is working as a statolith in Clostridium, where it senses the gravity. These are on the surface of the water body. They are planktonic. For them it is very important to be on the surface because they are performing photosynthesis. So anywhere where they can sink, they need to reduce what they store in the body so that they will become light and float on the surface. So who will sense the gravity in the Clostridium? The gypsum crystals. Because they are heavier than the surrounding, they will attach to the starch granules and they be denser than the medium. Whenever there is a gravity, they sink down in the semicell, in the vacuole. Then the organism will use up the stored food, make itself light so that it can float on the surface. This is planktonic character, floating on the surface. And the gypsum crystals in the terminal vacuole acts as statoliths, which will help the organism to judge the gravity as per its nutritional need to float on the surface of the water. This is all about gypsum crystals.